Hey there, friends, and welcome to episode 286 of Just the Zoo of Us. This week I am joined by an evolutionary biologist and herpetologist for a look through the magnifying glass at miniaturized animals. Our guest studies species who have shrunk down to incredibly small sizes, like frogs and chameleons who could gather on the tip of your finger. And he has some fascinating insights into why and how animals get so darn itty-bitty, and what could be stopping them from getting any itty-bittier. We discuss silliness in scientific names, assuming a spherical frog in a vacuum, chameleons called bee-nana, whether jawbones spark joy, how many brain cells you really even need anyway, and so much more. Despite the tiny scale of the subject matter, we cover a lot of ground in this conversation, and I think you're really going to like it. Just the Zoo of Us presents a review of the animal kingdom in miniature with Dr. Mark D. Scherz. Hello, everybody. This is Ellen Weatherford with Just the Zoo of Us, your favorite animal review podcast. I'm so excited this week to have a brand new friend here to talk about some of the little guys, some of the tiny little friends that might sneak past you unnoticed. But let's take a magnifying glass down to the forest floor and check out the little guys. This is Dr. Mark D. Scherz. Say hi, Mark.
Hi, Mark.
It's every time. It's every time. I need to stop. I need to come up with a better way.
No, I think it's perfectly adequate. It's those of us who are longtime listeners of various podcasts who are familiar with this particular opening.
I need to stop painting a giant Looney Tunes target on my chest right at the beginning of the episode.
I mean, it was semi-autonomic in my case. It just kind of like, oh yeah, she said the thing, I have to do the thing, so...
That's true. That is ingrained into the dad consciousness. Like, yes. As soon as you have a kid, that neural pathway just becomes cemented in.
Yes. Hello, hungry. I am father.
Well, Mark, you have studied some of the most charming, dainty little creatures in the world that I think a lot of people would be fascinated to hear about the journey that you have taken to work with these little critters. How did you get into the work that you do with tiny animals?
So it all goes back to when I was about 8 years old, and it's actually before I was 8 years old.
This feels like a therapy session.
Yeah, it was so brace yourselves. The story is long, but essentially what happens is, because I can't remember it from the first person perspective, I can only like look back at the things that I produced at those young ages, and I can see what was kind of happening. So around the age of 7, I made a book in school, like first grade, where apparently there were these two otters who went on a trip. I lived in Ohio at the time. When I was small, I lived in America. And these otters went on a trip from Ohio to Madagascar and did all kinds of wonderful things in Madagascar. It was like 4 pages long, right? So I was 7. And in any case, that is the first evidence that an obsession had formed with the island of Madagascar. And it's not really clear where that came from.
It was prescient. That was fate speaking through you.
So there's been a strong connection to Africa in general through my family history. All of my grandparents were missionaries, and my mother grew up in Malawi. So there's always been kind of like Africa. There's a lot of our African stuff at home. I went to Malawi when I was a small child to visit my grandparents and those types of things. So in any case, there was always this kind of like connection with Africa, and somehow that kind of got sidetracked, kind of missed the continent and went into the Indian Ocean. There might have been some connection with some, episodes of Steve Irwin or Mark O'Shea's adventure, which, although I think that was later, where he also went to Madagascar. So something happened, at which point I became completely obsessed with the island of Madagascar from around the age of 8 years old.
Correctly so, I should say. It's a fascinating
island. Valid. I totally don't question my decision whatsoever.
I was right.
Yeah, definitely. Like, no question. In fact, I'm kind of questioning literally everyone else. But in any case, it was Madagascar, the island, and its wildlife that kind of got me first. And then progressively, I made my teachers' lives in school a living hell because I didn't want to do anything else except for focus on the island of Madagascar. And so what I would consider very good teachers made accommodations for that. So my French teacher went out and brought a documentary from the library about Madagascar in French so that I would be convinced that I should watch things in French. Those kinds of things, right?
That's proactive.
Good teaching.
You know what
right?
I mean?
That's
is
really good.
Tie in, make it relatable and engaging.
Especially when the student literally won't do anything else and will just sit there fiddling the whole time, if not, because they have attention problems.
That's your Roman Empire. It's all Madagascar, baby.
Absolutely. Literally, I would just sit there and write on the side in the margin of my paper, what are the names of Latin names of species from Madagascar that I can remember? So in any case, after many years of nagging my father, I finally went to Madagascar for the first time when I was 14 years old, because by that time he couldn't put up with it anymore, and he also wanted to do one big adventure with me. So we went on a tour with other reptile-interested people, and because by the time I was 14, it had become really an interest in reptiles. I'd grown up around reptiles and amphibians a lot. I was living in Ohio. There are a lot of amphibians there, and quite a few reptiles. So, my father would take me fossil hunting. He would pull out the slab of shale, I guess, from the creek. He would look at the fossils, and I would be like, "There's a salamander behind here."
Wow.
So that was kind of that was my young childhood, and those were really the impressions that I grew up with. So without realizing it, I'd become very channeled and focused on reptiles and amphibians. Why is this relevant? It's relevant because I'm not just interested in miniaturized animals, but I'm also the curator of herpetology at the Natural History Museum of Denmark. So I've come into this position of working mostly on reptiles and amphibians, and that is the angle through which I have come to the tiny animals. To get to the tiny animal part of the story, we have to continue from where I left off. So I went to Madagascar that first time. I was 14, and at one point we found in a bush in the rainforest, we found in the top of actually not a bush, a tree fern, someone pulled out this unbelievably tiny frog. Unbelievably tiny frog. And I remember seeing this animal and being like, what is that? That is...
how'd you get like this?
It's smaller than most of the spiders I've seen. What has happened here?
Who's running amok with a shrink ray in the rainforest?
Yeah. And then, to go off on a tangent, we still don't know what that is. I have photographs of it. We still don't know what species it was.
Wow. What a mystery.
And now I'm an expert on the reptiles and amphibians of Madagascar. I should know, and I still can't really tell you what it is. I think I might know what genus it's from, but I'd have to go back and find another one to really confirm. In any case, so already on that first trip, that was it. It was focused. And from then on, I came back, I did high school, whatever. I went off and I did my bachelor's degree in Edinburgh in zoology. I really wanted to do my degree already in herpetology because I knew what I wanted to do. I was one of those awful kids in school who's like, why am I sitting in a history lesson? I don't want to be in a history lesson. I want to be, studying the things that I care about.
There are far worse things a teenager could be obsessed with.
Definitely.
Honestly, you win the jackpot.
This was an actionable thing. It was also very heavily incentivized and supported by my parents. And so I wound up going to Edinburgh University in Scotland to do my bachelor's degree, which was really great because unlike, American colleges, British universities don't have very many general, if any, general courses. So I didn't have to do anything except the things that I cared about. And as the courses got more specific, it was really ticking the right part of my brain. Moving ahead, at the end of that, I was like, okay, what I really want to do is study reptiles and amphibians of Madagascar. And so in my third year of the 4-year course, I wrote to the world experts, Professor Miguel Vences from the Technical University of Braunschweig, and Dr. Frank Glav from the Zoologische Staatssammlung München, the Zoological State Collection, or Bavarian State Collection of Zoology in Munich. And I wrote to them an email that was like, I really, really want to do a PhD as soon as possible. This is my idea for a PhD project. Can I do a project in your lab? And I got a response, I think within a day, where Miguel wrote back to me and he said, I've just come off the phone with Frank. Although we think your enthusiasm is great, first of all, it's not possible for you to do a PhD without having done a master's. And secondly, we don't think there's any possibility to get any kind of funding like this. So if you can find somebody else to co-supervise, then maybe, but otherwise, no. And I went home and sobbed for a while.
A gentle letdown, but a letdown nonetheless.
Exactly. It was incredibly soul-crushing. I eventually found out that I could, in fact, do a master's with Frank in Frank's working group and come to Munich to do that in English, which was very beneficial, although I speak German because I actually grew up in Switzerland after I lived in America, and my family is German. So I wound up going to Munich and doing that master's there and continuing on to do a PhD. While I was doing that, I showed up and I was like, look, I really, really want to do taxonomy. I really want to be involved in species description, really interested in species discovery. I love Madagascar. By that time, I had been to Madagascar 4 or 5 times because during my bachelor's, I was running my own expeditions to Madagascar, organizing and running them through public funding. And so I went to Munich and I said, look, I've got this, this is the overview. This is what I really want to do. And Frank was super supportive and it was awesome. So I got to work with the dream people in the end. So Frank initially and then Miguel also. And as I say, they are the world experts. They wrote the book. my Bible, the field guide to the amphibians and reptiles of Madagascar.
I feel like when you're really, really interested in hyper specific topics, it can be interesting how you elevate certain figures in those fields to A-list celebrity status.
Definitely.
And you're like,
Definitely.
Man, boy, I hope someday I'll get to cross paths with this person. And you can literally just send them an email.
Yes. Yeah, literally. Yeah. But on the other hand, I have had people come over to me at a conference sobbing about how wonderful it was that they just got to meet this person that they've been reading the papers of for the last 20 years. They might have been, mildly inebriated, but anyway, they were still very, very enthused.
Because that's how it feels, right?
Yeah, so when I started working with Frank, I said, hey, I would like to work on Bufus, really beautiful tree frogs from Madagascar. And he said, what about rhombophrene, these really ugly terrestrial frogs from Madagascar? And I said, all right then.
I'll take what I can get, I guess.
Exactly. And that was definitely the right call, because what I wound up working on were these very poorly known, what are called narrow-mouthed frogs. The Microhylidae is the family. And while I started on that, just a year before, another PhD student in Miguel's lab in Braunschweig, her name is now Dr. Andulalau Rakutuarison. She's Malagasy. And she had just started also working on cophyline microhylids from Madagascar. And her group of main focus was a genus called Stumphia. And the Stumphia are stump-toed or dwarf frogs from Madagascar. And so during the course of my study, I was increasingly working with her, and we wound up publishing together a revision. This was one of the big chapters of her PhD, but I was second author. We worked very closely together on this paper, describing 26 new species of dwarf frogs from Madagascar. And during that work, it became clear to me how phenomenally diverse miniaturized frogs are. I mean, this is the fact that you can describe 26 new vertebrates in a single paper. It really attests to the fact that these groups have been heavily overlooked. They've been taxonomically poorly understood. They haven't even been accessible in many ways until now. The last big revision on Stumphia was published in 1992. And so we were like, okay, this really blows everything else out of the water when we're able to apply the breadth of methods that have become available. We're taking genetics, we're taking bioacoustics, recording these frogs in the field, and able to integrate all of those data to produce what wound up being a monograph. It's like a little book on these frogs describing 26 new species in one go. And as we were working on that, it also became clear that there were several other genera that had been confused with Stumphia previously that look like Stumphia. They look exactly like Stumphia to the naked eye, especially to the non-expert eyes. So until now, everyone's been treating them as Stumphia, but genetically, they're completely different parts of the tree. And what's happened there is they have converged on extreme miniaturization. They've gotten basically as small as it's possible to get as a frog. And when you do that, as it turns out, and this was something that really started getting my cogs turning and I got really, really excited about, and now it's the main thing that we're focusing on in my lab, is the realization that when you get small, you tend to converge morphologically to this kind of morphology that makes you really, really hard to tell apart from other things. And so I wound up having this opportunity to describe a new genus of which no species had been described before. And I'm very fortunate to have had a very good success with the press release for that paper, although I guess I do take some of the credit because I managed to lobby for and convince the co-authors to name the species in the genus. So we named the genus Mini, and we named the 3 species in the genus Minimum, Minuscule, and Miniature. which because I'm a big
banger.
Fan of. I mean,
the fact that you can get like 3 puns in one go.
It was deeply satisfying.
Like to get 1 pun is exciting, but to get like 3 in one, that is like you really have to stick the landing on that.
It was, I think, the greatest scientific achievement of my career has already happened, and it is that. And so everything that I do after there is just kind of bonus content.
That's your immortality.
That's my legacy. Exactly. And that is also what I'm mostly known for on the internet is having done these silly things with the frog names.
I've been telling people, like taxonomists, I'm like, have more fun. Be
for
more
sure.
silly.
For sure.
Because then people are going to think it's funny and they're going to write more articles about your discovery.
Yeah.
Like, be sillier.
Exactly. I can give you so many good examples of that. I don't know if you've already had an episode on silly taxonomic names, but I strongly suggest it because I don't know if you've heard of the extinct parrot, Veni Vidi Vici. There's the jelly bean and jelly belly beetles.
Oh, I did a video one time on a bunch of scientific names that were named after Pokemon. There's a lot of them.
Yeah.
A lot of them named after Pokemon.
I've named species after, I've named one species after Fangorn Forest from The Lord of the Rings. I'm a huge fan of Tolkien, so we've got one of those. We just named, last year, we named several new species of tree frogs. I got to work on tree frogs in the end. We named several new species of tree frogs after Star Trek captains. So, we're having fun. We're definitely having fun.
Have more fun with it.
And we're not the only ones, especially among the miniaturized people. People like to do silly things. Notably, there's a series of miniaturized salamanders where the genus is Thorius, which is a fantastic name, by the way.
That's
Thorius.
very cool.
I tried to lobby to name my son Thorius, but it was vetoed. But in any case, Thorius, and then there are several different species names that just rhyme with Thorius. So they're like Thorius aureus and Thorius arboreus. Those
Thorius
kinds of like
Gregorius.
Yes, that exactly. Yeah, it sounds like nonsense.
I like the Amelia Bedelia approach to naming species.
Yes, exactly.
I also feel like in another life, Tolkien was a taxonomist. He would have loved that.
For sure. I mean, as a philologist, he's essentially a taxonomist anyway.
Rest in peace, Tolkien. You would have loved taxonomy.
I actually think that he didn't like it very much, but he had some strong views on how science and like that it took the beauty out of nature.
Some of those chapters feel like species descriptions a little bit, because I'm like...
Yeah, but so much more impassioned.
We've been talking about these trees for a long time.
I mean, your listeners did come for an analysis of Tolkien's language and his tree descriptions, right? Because that's what we're into now.
Honestly, okay, the briefest possible tangent on Tolkien's tree descriptions.
Oh, yeah. Doesn't have to be brief.
In our wedding ceremony, we had a poem that Tolkien had written to his wife. And it was just a beautiful poem, but it was all about like seeing them as 2 trees with roots that had grown together and stuff. It was like extremely on brand. It was a very on brand Tolkien love poem, but it was about like lovers as trees, like with roots together. I was like, this is a banger right here.
Yeah, Tolkien's love for his wife bears no comparison.
What truly, a wife guy.
What a man. Yeah, what a wife
He's
guy.
He's like, I love 2 things.
Yeah.
Trees and my wife.
Honestly, that's a pretty succinct analysis. If that hasn't already been published...
Well, you gotta make space for his other true love, the homies.
Yeah.
Hug your homies.
The boys, yeah, definitely. Predominantly the boys. So in any case, I guess I can wrap up my story there. I got incredibly deeply interested in this whole process, everything behind the evolution of extreme miniaturization. And then I just made it my entire personality. And so what I did last year, the year before last, so 2023, I broke myself a little bit and I applied for what is called an ERC starting grant. And it's the European Research Commission, European Research Council, European Research C, an ERC starting grant, which is like the most prestigious research grant in Europe. I feel sick just saying it. And then I got the darn thing, and now I actually am committed. The next 5 years in my lab, we are using comparative genomic techniques to produce reference quality genomes of the world's smallest vertebrates to find out what on earth is happening, why they are the way they are, how they got the way they are, and what is making them not get any smaller. That's such an interesting question. The best thing about this is that everyone asks the same questions as I do. When I bring up these topics, the question that I get asked is like, why don't they get any smaller? And why are they the size they are? Like, how did they get that way? And I'm like, I don't know. Let's find out. Let's do it.
You're like, that's what I'm saying. Why
That's
are they
what
like
I'm
that?
saying. And apparently the ERC people were like, yeah, you can have 1.5 million euros and try and answer that question. And
They're
I'm
like, yeah,
okay.
dude, why are they like that? Figure it out.
Yeah. So that's my goal. That's what we're doing in my lab for the next 5 years. And hopefully beyond that is really trying to understand, like, how did we get like this? And like, why is the smallest hummingbird, the smallest dinosaur that has ever lived, exactly within like a gram of the smallest mammal in the world? And not just that, both of the smallest mammals are within a gram of each other. And in terms of body length, the same size.
So it does seem like they're like asymptotically approaching some sort of critical mass.
Yes! And actually, the best part is we know why. In the case of endotherms, we know what it is.
Hi there, quick editing note. I realized we used this word a few times without explaining what it means. Endotherm means what you may have described as warm-blooded, as in an animal that maintains its own body temperature, mostly mammals and birds. The opposite is ectotherm, which are animals that rely on their environment to keep their own body warm, like reptiles and amphibians and fish. So when you hear us use those words, that's what they mean. Back to the episode.
If you have to make your own body heat, you have to get the energy for that somewhere. You are a little furnace, and you need to keep putting logs in the furnace. And in order to get the logs, you have to fly about or run about, and you need to get it from somewhere. Hummingbirds crack the code. You get sugar, pure sugar, that's just rocket fuel. But there's only so small you can get before you can't get it as fast as you burn it. And so actually there's a physical limit on how small you can be as a free existing endotherm. And that is why all three of those groups, there's a beautiful equation on this in a paper from 1996 that shows that it's basically, it literally is an asymptote. You cannot get smaller because you have to consume infinite food per second. And so, you know, and the way that they overcome that, of course, is many of those animals, they undergo torpor. They, instead of sleeping, they put their body into some other kind of rest that doesn't burn the same amount of energy. And so they have these temporary solutions that have allowed them to get even smaller. But basically, it's, as far as we understand, practically impossible for an endotherm, as we see the endotherms today, to exist at a smaller size than the Etruscan shrew, the bumblebee bat, and the bee hummingbird.
It makes sense that as they're getting smaller, so too is their fuel tank. Like,
yeah, right.
They don't have the capacity for storing as much fuel as they would need as they get smaller.
Yeah, right, exactly. How do you carry the fat? Well, now you've got to lug your fat with you everywhere you go. You can't really be storing any fat because you have to go and take it with you as your luggage. Can you imagine? You have to like stuff your whole suitcase full of hamburgers, and you're like eating all the hamburgers at the same time. You're like, okay, well, I can only stuff so many before it's not a return on investment.
But you have to eat a hamburger every like 10 minutes.
Exactly. Otherwise you die. And of course, you know, it's a bit different for the juveniles. There must be something else that's going on. But of course, you know, juvenile metabolism, adult metabolism was quite different from each other and those kinds of things. So for the endotherms, it's really interesting and relatively well explained and well understood. For the ectotherms, no idea. And that's where things get really interesting because, as you mentioned, asymptote, there's this weird clustering thing. And I like to think that I'm one of the few people who've really noticed how extreme it is in the frogs and fishes, because both of them have a surprisingly large number of independent lineages that are around 10 millimeters adult body size. Like, there are at least 20 different frog lineages, and probably substantially more, that are all that size. And over, you know, hundreds of millions of years of, or at least tens of millions of years of separation between those different things. And then the fishes, it's the same length, which is very weird.
That is weird.
Isn't it?
Like that's the sweet spot.
Yeah.
That's the sweet spot for like, you want to get this small, no smaller.
Exactly. So the smallest fish in the world is Petacypris progenetica, although, you know, it's always a fight. It's always a fight. There's also Schindleria brevipenguis, which is also very, very, very small. And so these things are around 9 to 10 millimeters adult body size. I would have also thought that there were fish that get smaller. Like, it feels like there should be fish that get smaller. But I would point out that by mass and by body volume, they are much smaller than the frogs, because fish approximate a little rectangular prism, whereas frogs approximate a sphere. Yeah?
Assume a spherical frog.
Yeah, exactly. What is it? Spherical frogs in a vacuum. That's how we make all of our models.
You could take like a 3D model of a sphere and then just map a frog texture onto it.
Rain frogs have really optimized. For physics models, all you need is a rain frog. Anyway, that's a different tangent. So one of the really important things that matters here is physics. I hate physics. I failed physics in school. I'm terrible at physics.
I already don't like it.
But some of the relationships are actually really simple, and in fact, they're not physics, they're geometry. So we can kind of be like...
Geometry's on thin ice.
Geometry, exactly. It's like right on the edge. But one thing that we do know is the cube rule. If you double in size, you increase cubically in volume.
Oh, I was just talking about this like a few episodes ago when we were talking about ants.
Right.
Like, that's why like a giant ant wouldn't be super strong.
Yes, exactly. And what that means for a vertebrate is the skin. Amphibians and fishes use their skin to a significant extent in oxygen exchange. It also is critical for osmoregulation, regulation of your water, and regulation of all the minerals that you... Hygroregulation, let's say, is the most important thing. When you decrease in size, you desiccate so much faster. Not just like proportional to body size, but like exponentially faster.
Is that because of like the change in proportion of like surface area?
Yeah, your relative surface area is enormous compared to your volume, right? If you get bigger and bigger and bigger, an elephant has actually really small surface area relative to its body size. But a tiny frog has an enormous surface area relative to its very small volume. And because of that, there's a lot of pressure to lose water from inside the body. And I believe that's the reason we don't see any of these miniaturized frogs outside of the tropics. You cannot have an animal that loses all of its body water, as soon as you expose it to the air, because it will die. It can't exist.
Right.
And so I think that there are environmental parameters, there are these physical parameters, and then certainly there will be genomic parameters, both associated to those and intrinsically, that are regulating these kinds of things as well. I can really go on this topic forever. Like, we can talk about the number of neurons you need for a brain, and how many neurons does it take to make a vertebrate heart, and do you actually need to have blood flowing through your teeny tiny fingers if you can anyway exchange oxygen over your skin? Super interesting topic. So and literally we could talk about it for hours. So maybe I hand it over to you and you ask me relevant topics, because otherwise.
Well, what I was wondering is because, we were talking about the limitations to how small you can get. Like once you reach a certain size, it is going to harm you to get any. And in some ways, it sounds like being really small can be harmful. Like if you're losing moisture and losing heat and losing temperature to the surrounding area. So
plus you can be sucked right up a straw. I mean, just it's
so why do it at all? What is the pressure that pushes them towards being small?
Okay, yeah. So what's the positive pressure to become smaller?
Why small?
Yeah, why so small? Important question. First of all, I will just pull the I don't need a reason card, which is drift. Body size changes according to genetic drift, according to body size. Drift, it is a semi-neutral trait, and under certain circumstances can certainly evolve in a neutral manner. And as a result of that, you will see drift in body size. And so over time, of course, that drift can develop kind of a direction through successive selection events. But on the whole, we assume often that it's just evolving in a Brownian motion kind of way. So things can get very small by chance.
I like the idea that we just weren't paying attention.
We
got small and we didn't notice.
It's like, oops, I just honey, I shrunk the kids by accident. Over 100 million years.
I shrunk the kids, and they shrunk their kids, and they shrunk their kids, and now we're too small.
Right, exactly. So it can happen essentially by chance, but then it gets maintained, right? We see these things through to today. What is the benefit? And here is where I think we start having to pull apart the different groups. Some of these things will be general, and some of them will be specific. What is the benefit to a frog moving from a terrestrial, on top of the leaf litter frog, into a super tiny mini frog? I can see so many benefits. I'll bore your ears off, but some of them are really super obvious, and you can think of them yourself. So, food availability. What is the relative yield if I eat, let's say, a springtail, and I'm a big frog? Low, really low. the smaller I get, the more food per my body mass I get from that springtail. And anybody who's been to a forest and turned over a log knows that springtails are hyper abundant in the leaf litter. Like, there is no shortage of food in the leaf litter. The larger you are, the less reward you get per springtail. When you get small, your relative yield gets high, and you can maintain a population based entirely on springtails. It's not a very diverse diet, but it would exist. Plus, you're maybe less appetizing to a larger-bodied animal, like a big snake. A big snake is super happy to eat a big frog, but not so keen to eat a tiny frog, so you can escape predation pressure. But I think the real key, and I think there was, I can't remember who it was, but there was a paper in 2012 where this was put forward as an idea, is when the frogs miniaturize in particular, they start to explore a space that is not available to a larger animal. And it's what I call the interstitium. It's this interstitial layer between the soil and the surface of the leaves, which in a complex rainforest is often, between 5 and 25 centimeters of three-dimensional space that is like honeycombed chambers of dead leaves. That is an enormous surface area that is only accessible if you can walk between it. And one of the cool things that we see convergently evolving in these miniaturized frogs is that they stop jumping and they start to walk slowly instead. So they do this kind of slow motion prowl. And when you stop hopping, it stops being a problem that your three-dimensional space is limited on top, and you can kind of crawl through those very narrow spaces. And so I think that this has been a really key thing, and that may be the explanatory factor for why we have quite so many independently miniaturized frog groups, because in every case they are in the leaf litter and they're really among the leaf litter. Many of them even call from inside the leaf litter.
They've unlocked a whole different map.
Yeah, exactly. You unlock,
that wouldn't even register to me as a location.
Yeah, it's a whole niche. I mean, all of the entomologists know about this because that's where you search for the goodies. But when you're a vertebrate biologist, you're like, okay, I see it hopping on the leaf. We dig traps, and that's how we find a lot of these things. But in fact, what we started doing, especially back in 2016, we started doing this a lot, is we'll go to an area, we'll take 1 square meter of leaf litter, and we'll take a big rice bag, and we just shovel it as fast as possible into the rice bag, carry the rice bag back to camp, and then delicately dissect the rice bag, take apart all of that leaf litter, because that is where we find the pygmy chameleons, that's where we find the pygmy skinks, we find the pygmy frogs living inside that cluster.
They load it into the bug server.
Yeah,
like,
exactly.
I'm gonna join y'all's
it's
map. You seem like you have something going on. The Bugiverse.
Yeah. So, Ant-Man is very enviable to me. So whether or not a similar kind of thing is happening for the fish is hard to say. We know that there are a lot of reef fish that live, specifically miniaturized fish that live inside the crevices created by corals. Again, exploring a physical space that is not available to a larger animal. So there's that physical space aspect to it as well. So loads of different things. In every case, they have also different explanatory factors. It's a super complex thing, but it's interesting that it's so widespread. I should also point out that we thought that there are hard limits on how small a vertebrate can be. And we thought that for frogs it's around 8 millimeters, and for fishes it's around 8 to 10 millimeters. And then 2 years ago, I think it was now, they described Brachycephalus pulex from South America, I believe from Brazil. It's a flea toad, which is a member of the pumpkin toad group, Brachycephalus. And it's, I think, 6.5 millimeter adult body size.
A pioneer.
Which is truly sickeningly small.
An innovator.
And
changing the game.
It really is. It absolutely is.
Disrupting markets.
It pushes that limit that we thought existed down by 2 millimeters, which 2 millimeters sounds like nothing, but when you're at that body size, that's 20% of your body size.
Highly recommend Googling this little guy.
Yeah.
Because it's baffling. This
Brachycephalus pulex, yeah.
I mean, it's like a molecule of a critter.
Truly, I got to, so I had a journalist bring that paper to my attention. They were like, hey, we'd like you to comment on this. We know that you like mini frogs. And they sent me this before it was published, and I was like, no. No way. I don't believe you. I can't believe you. Like, everything we know about frogs says this shouldn't be possible. And then here it is. And I absolutely believe them. I think they're absolutely dead on that this is an adult frog. Because that's always the debate. Are these adults? But I'm sure that they're right. I'm sure that these are adult frogs. The analysis they did in that paper is simply fantastic. And it breaks the mold, absolutely, because it goes down by a substantial margin. So what on earth are they doing differently that the other ones aren't?
When you mentioned that this was a member of the pumpkin toad family, I was also thinking about pumpkin toadlets, because when you were saying that when frogs get to this size, you start to see them stop hopping.
Oh yeah,
and that's something I remembered from our pumpkin toadlet episode, that like they still try.
Yes.
They're like, I'm trying, very bad at it.
Yes,
like they're too little to jump right.
Truly horrible at it. Now, this is so I haven't listened to that episode, so I don't know what was said on it. So let me opine on that topic, because I've now repeatedly, I think 3 times in a row, I've had a grant rejected. So my goal for my lab is to gain a holistic understanding of miniaturization, not just at the genomic level, but using the genome now that we've got the funding as a kind of a foundation to understand miniaturization in vertebrates from every angle, from the physics angle, from the cellular angle, from the neurobiology angle, and from the locomotive angle as well. And so I put in a grant to replicate the study on the pumpkin toadlets, but not on pumpkin toadlets, but to do it instead in the Malagasy miniaturized frogs, which are not at all closely related, but which have also a very slow locomotion. And the reason I put that in is because I have worked for a long time with miniaturized frogs from Madagascar, and I know for a fact that a 8 to 10 millimeter long frog from Madagascar can jump for a meter, 100 times its own body size, and then jump again instantly. So that is not possible for a brachycephalus. And I believe that the reason for that difference is that brachycephalus are broken, and this is not a factor. It's not miniaturization. I don't buy the explanation that the authors have. I think that something has happened that has broken possibly the inner ear, but some other mechanism possibly as well, for brachycephalus in particular. And that something else is going on in the rest of the miniaturized frogs. So that hasn't happened in the other miniaturized frogs. The corroborating piece of evidence that I see for that is that brachycephalus, not only can they not jump properly, and by the way, when they jump, it's not like they're completely failing to launch. They launch just fine. Looks great when they launch. It's in the air that things start to go wobbly.
They had a plan for getting up, not for getting down.
Yeah, exactly. This plane was meant to lift off, not to land. And so they're perfectly good at that part of the arrangement, and then things go wild. But the other thing that we know has happened with brachycephalus is that their inner ear complex is simply broken. They can't hear either. Like, they can't hear their own advertisement calls.
Right. Yeah, I remember talking about that.
It's bonkers, right?
Yeah.
And again, although this could well be a consequence of miniaturization in their specific case, it's, I think, highly unlikely that is a rule, that is a thing that must arise due to miniaturization.
You guys just did it wrong.
Exactly. And the way that it was put in the paper talking about the pumpkin toadlets was that there is a physiological limit to how small the semicircular canals of the inner ear can be in a frog where the fluid dynamics breaks down. And the alternative, and to my mind, much simpler explanation, is that these frogs have simply got it wrong. They might have, for instance, a slightly too viscous fluid inside those inner ears, and then it doesn't work either. Or whatever, right? So, because we know that it works in the other things. The thing is that we don't yet have proof that it works in the other things, because no one has taken a slow motion camera out there into the field to do it. And so I keep trying to apply for the slow motion camera, and I keep getting the grant rejected with totally rubbish feedback. So
listen, they can't all be wire frog small.
They can't all be winners, yeah.
I do love the explanation of this one's just broken. Like, I don't know, something went wrong.
I think that's also just such a wonderful thing to highlight. Is like, one thing that miniaturization does do, almost as a rule, is that it breaks things. And that is in itself, I think, the biggest selling point of my grant. Possibly the thing that got people most excited is when you get small, things get reduced. At some point, you have too few cells to make the thing that you're supposed to make, so you kind of make do with what you've got. When you get bigger again, you don't necessarily have all those pathways that were originally making the original structure. So you need to kind of innovate in order to make the thing that you should have, or you now have a new opportunity to re-innovate. So the way that I put this is that I think miniaturization as a process is a potentiator of novelty. And the cool thing is, if you look back in the fossil record, paleontologists have been talking about this for quite a long time. They started to talk about how becoming very small was an absolute key innovation for dinosaurs to be able to develop powered flight. It was also, by the way, strongly connected to this thing that happened in our early ancestors, where they took jawbones that used to be in the jaw, and they were like, we don't need them for the jaw anymore, we're going to put them in the ear. Those kinds of changes are the things that we see, or we can observe, or we can predict to happen in hyper-miniaturized animals. So, what's likely, in my opinion, to have happened is a process of miniaturization that relieved the pressure, changed the relationship of the reliance on different structures within the jaw, freed up those posterior bones, and those bones could then migrate to become part of the inner ear, which is a synapomorphy of the mammals. And so, yeah, these kinds of dramatic transitions that we see within the vertebrates could very clearly be linked to miniaturization in our past. And you go beyond vertebrates, and you see the same thing. It turns out, tardigrades, everybody's favorite water bears, tardigrades are miniaturized worms.
Oh.
They are, in fact, worms that have somehow lost the middle bit of the segments. They have the front bit of the segment, they have the back bit of the segment, and they're missing all the segments in between.
Didn't need those. Got rid of those.
And you can look at it there have been genomic studies that are basically indicating this as well. Evolution is a tinkerer, right? And one of the things you can do as a tinkerer is you can get rid of all of your tools and you can go out and eventually buy or make new ones. And I think that's what miniaturization does.
I do like the idea of miniaturization as like Marie Kondoing your body.
It is. It's good. It's a good thing.
Of just like going through and being like, you know what, this jawbone doesn't spark joy.
It doesn't spark
We're
joy.
not using it. Let's toss it out. And then later on, like a year later, you're like, oh man, I tossed out that jawbone.
Absolutely. And so you might still have the instructions to build the jawbone, but not the same materials. And so you might get it slightly different when you do. Or, a lot of these patterns are extremely conserved, right? So it might actually come back just the way that it was before, and you wouldn't ever see those signals necessarily.
Maybe by the time you need a new one, a new model has come out, and maybe now you get a better one.
Yeah.
So,
yeah.
Sometimes you got to throw things, you have to let things go.
Exactly.
Let things go so that you can build back better.
Yeah. And what do you think, a question to you, what do you think is the one item of vertebrate anatomy that we like to get rid of the most?
Oh, gosh. We get rid of limbs a lot.
Yeah.
That's a lot of times we're like, I don't need that.
Ding, 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 ding. Yes, perfect. Perfect answer. We get rid of limbs. And where do we start? We start with the fingers. And so this is a perfect example. In miniaturization, one of the first things that we start to see in frogs is they just stop making their fingers.
The fingers are already so little.
They're
like,
so small.
We're all little already.
Yes, and what is the difference between having 4 fingers and 3 fingers when your fingers are literally 0.5 millimeter long? None. Makes no difference at all. And we know this because there are 2 frogs that we named from Madagascar, Stumphia contumelia and Stumphia obscena, named for the fact that they've only got middle fingers.
I'm so glad I'm not recording video because the gesture...
Well, contumelia means contemptuous, and obscena means obscene. So they're named for the fact that they only have middle fingers.
Because you demonstrated what that looks like for me. That was a special visual just for me.
Well, it is in fact what happens in these cases. So in extreme cases, you go down to basically mitten hands that have a single functional finger, and the functional finger that is left behind in the case of frogs is the 3rd finger, but they've only got 4 fingers, and so it looks like a middle finger. And then for the toes, we see the same thing. And what's brilliant is that I think in the 1980s, there was this fantastic series of studies where they found out that not only do we see see reduction of digits in these different miniaturized things, but it's always happening in the same order.
Huh.
It's always the first toe, and then the 5th toe, and then the 2nd toe.
Those are your sort of peripheral, optional toes.
Well, more importantly, from a developmental standpoint, they're the ones that ossify last. So we lose things in the reverse order in which they are made, which makes a lot of sense.
Yeah, because you're like, I'm building all these toes. I already built 3. Maybe I just don't want to build the last 2.
Well, if we want to go into the theory, how do you get a miniaturized thing? You can do a lot of different things. You can speed up your development, and you can say, okay, I'm an adult now, even though I'm teeny tiny, and you wind up with basically a homunculus, perfectly proportioned miniature organism, right? Or you can say, I'm going to speed up only the genitalia, or only the reproductive organs, and then you wind up with what we call a pedomorph, and those pedomorphs, that just means that they look like a baby. And then there's progenesis is also basically shifting things forward in development. And so you have these, like, there are very well established and historically long discussed and disputed mechanisms by which you can, become miniaturized to begin with. Of course, it's actually a lot more complicated than that. But yeah, so you can kind of generate a very small body size through these different mechanisms, and we see that as well. So one of the frogs that always gets overlooked when people talk about the mini frogs is the other one that I described in the same paper called Rhombophrene proportionalis, which tells you already in the name what it is. It is a proportional dwarf. It is a perfectly proportioned big frog that is just 10 millimeters long or 12 millimeters long.
That's weird, right?
Isn't it weird?
You see, like, when they get smaller, they get kind of babyish almost.
Pedomorphic, right. You can even go so far as to have, at the same time of pedomorphy, you can have neoteny, and that's what the miniaturized fishes have done. Neoteny is where you retain the larval characteristics, but you develop your gonads. So think of this, the axolotl or axolotl. So axolotl are neotenic. They are not miniaturized, but they never undergo metamorphosis that they are capable of undergoing, but they start to reproduce without developing into those so-called adult or terrestrial salamanders. That process is only possible for salamanders and for fishes. It is not possible for terrestrial tetrapods. And because of that, we do not see it being connected to the terrestrial tetrapods, including salamanders, by the way. None of the smallest salamanders are neotenic either. But we do see it in the fishes, and it's actually basically a rule in the fishes that the smallest ones are neotenic. So I have some thoughts and hypotheses about why that is, but we don't need to go too deep into them now. It's just to say that is another mechanism by which you can, or that there are these different mechanisms that feed into how you can get to be so small in the first place.
Hi, friends. We're going to take a quick break to hear from our friends on the Maximum Fun Network. We will be right back with even more, so stay with us.
If you like too many podcasts, you'll love Soundheap with John Luke Roberts. It's got clips from all your favourite podcasts, such as Diary of a Tiny CEO.
Leonard Sprague, tell me how you make your money.
I go to the beach and I steal people's towels.
Remember Armour.
Do you remember the trend of everyone whacking themselves on the head with hammers and mallets when they wanted to lose weight?
And Elty John's Lobberly Songs.
I'm here today with Kiki D. Hello, Kiki D.
Hello, Elton.
There's dozens of episodes to catch up on, and brand new episodes going out right now. So, if you want far, far, far too many podcasts, then look for Soundheap on Maximum Fun. Boop boop.
All right, we're over 70 episodes into our show, Let's Learn Everything. So let's do a quick progress check. Have we learnt about quantum physics?
Yes, episode 59.
We haven't learned about the history of gossip yet, have we?
Yes, we have. Same episode, actually.
Have we talked to Tom Scott about his love of roller coasters?
Episode 64.
So how close are we to learning everything?
Bad news. We still haven't learned everything yet.
We're ruined!
No, it's good news as well. There is still a lot to learn.
Woo!
I'm Dr. Ella Hubber.
I'm regular Tom Lum.
I'm Caroline Roper, and on Let's Learn Everything, we learn about science and a bit of everything else too.
And although we haven't learned everything yet, I've got a pretty good feeling about this next episode.
Join us every other Thursday on Maximum Fun.
I would also, because you did mention in Madagascar, there are some delightful, teeny tiny, the teeniest reptile you can imagine. Can we talk about the teeny tiny chameleons in Madagascar?
Yeah. I also had the great fortune of being involved in the description of what is still currently the smallest squamate in the world, although the researchers working in South America will fight me on that.
Don't take my title, please.
There's active debate. So Brachysia nana, which only afterwards was it pointed out that when you abbreviate Brachysia nana, you get banana. So again, bring witticisms, although we didn't intend to in this case.
Bee Nana.
bee nana. So Brachysia nana is currently the world's smallest squamate, and that makes it the world's smallest amniote. For those of you doing your academic bingo at home, we are amniotes too. So the amniotes are us and the birds and the reptiles in general. So Brachysia nana belongs to a group of Brachysia chameleons in the subgenus Evoluticauda, which is a name longer than the chameleons themselves. and they are extremely, extremely small. Brachysia nana was discovered on a massif in northern Madagascar called Sorata. I was not involved in the trip that went to discover that species, but it is a remarkably small chameleon. Really, truly remarkably small.
However small you think it is, it's smaller than that.
Yeah, it can very happily sit on the tip of your thumb with room to spare. When I talk about mini species and some of the other very, very small frog species, I like to talk about how many of them can sit side by side on your thumbnail. And bee nana, you could probably fit 3 or 4 on your thumbnail because they, like the fishes, they approximate to a rectangular prism rather than to a sphere, like the frogs, which take up then
You have
more
the
area.
infinity gauntlet of chameleons.
Yeah. So what's really interesting about Evoluticauda, these tiny chameleons, other than the bajillion things that are hyper interesting about them, is that they're spread around mountain tops in northern Madagascar and eastern Madagascar. And it basically is a relationship of one mountain, one chameleon. There is one species that is more widespread, but for the most part, they act like little island creatures. And that reflects another extremely important thing in why are there so many miniaturized animals. When you get very, very small, unless you can still fly, your dispersal capacity is highly limited. It's directly correlated, in fact, with how big you are. A big cat has a larger range than a small cat. And the same thing is happening with a frog. A large frog has a larger range than a small frog and a larger dispersal capacity. The vagility of the species, or the cruising range of the species, is related to its body size. And so we think that because these things are very bad at dispersing, and I can tell you chameleons are particularly bad at dispersing.
They're slow.
Yeah,
They're
you can
not
put
real
one of
quick.
these well, yeah, they're really astronomically slow, geologically slow almost. Like you can put one on a table and you can come back 2 days later and it's still like at the table. They're not going anywhere in a hurry. But when you have animals that are this small, a geographical barrier, like for instance a river, even a small stream, is a major barrier. And then we start to see other things that are dividing the species. Maybe there's just more gene flow on one side of the river and the other side of the river than across the river, and so you generate species. And so body size turns out to have an absolute relationship with dispersal capacity and therefore also with your rate of speciation probably. You have a higher rate of vicariance, of being separated by some kind of geological barrier, and therefore also a higher rate of allopatric speciation.
They reach like a tiny little like puddle stream and
Yeah.
they're like, this is the edge of the world. It
It's
goes
the
no
end.
further.
Absolutely. Yeah. And in fact, there's this other thing. So optics, another really interesting topic about these tiny animals. And here I would highlight chameleons, these tiny, incredibly ridiculously small chameleons, less than 2 centimeters long. They have a ballistic tongue like every other chameleon. They never miss. But one thing we know about eyeballs when they get that small is that they can't focus on infinity anymore. So in fact, they probably see a bleary, far-off world, except right in the near space where they have highly precise, incredibly acute triggering mechanisms for their ballistic tongues. And I will also mention the fact, I don't know if your listeners are aware, there are a huge radiation of salamanders that also have what we call ballistic tongues. And the smallest salamanders in the world likewise have ballistic tongues, except that they're salamanders. They have a much, much higher missing ratio. They hit the target way less often.
They're bad
Salamanders,
at it.
and yeah, if you watch some salamanders and frogs eating, you know that salamanders are, well, anyone who's had an axolotl at home as a pet is probably pretty aware that they're like pretty bad at being alive.
Yeah, maybe not a ton of processing power in that old noggin.
Not really. And that's where things start to get really, really crazy also about salamander miniaturization, because the smallest salamanders. So salamanders have gigantic genomes, huge genomes. Some of the largest genomes we've ever seen are salamander genomes. And one thing that correlates or directly with genome size is cell size, which means that the smallest salamanders in the world are around the same size as the smallest chameleons in the world. So also about like 18 millimeters, maybe a little bit less. They have proportionally far fewer cells because their genomes are 10 times bigger. And consequently, they get the moniker or the title of probably having the fewest brain cells of any vertebrate.
Oh, I mean, that's not a title you want to have, but...
Isn't it cool, though? Because what a cool system to go and study the fundamental limits of what do you need to have in the brain in order for it to do its thing. And how much of that is salamander and how much of that is fundamental vertebrate. Right?
We just recorded an episode where I did the research and presenting on box jellyfish.
Oh, yeah.
And how, we always roast jellyfish for being like barely even counting as an animal, like just barely skirting being alive.
They're weirdos. They push some definitions.
It's just a sack of water, so like we would always kind of go to jellyfish as our go-to, like the dumbest animal you can have
roast
while it
them.
still counts as an
Except
animal.
There's nothing to roast because they're barely alive.
They're empty. But one of the things I came across in doing notes for box jellyfish was that there were these researchers who were able to exhibit learning in a box jellyfish.
You can train them.
That immediately blew my mind. I was like, you're kidding. Like, how little brain do you need to work as a brain?
I saw a talk on this. It blew my mind.
Yeah, I was like, which makes me want, so
Absolutely.
Having already like read that, it had my mind already blown about like how little brain you need to function. Yeah, I mean, I buy it. Like,
for sure.
How few neurons can you skate by with?
Absolutely. Can I I'm gonna derail us for a second to talk about this because we talked about loss. And now we're talking about how much does it take? How few neurons do you need? One of the things that has happened in the world's smallest animals is extreme losses. In the case of the salamanders, there's this crazy thing that these hyper-miniaturized salamanders, important to know, salamanders and frogs and caecilians have nucleated blood cells. In fact, mammals are relatively unusual in having anucleate blood cells. But when you have to put a nucleus that is absolutely ginormous into a blood cell, it's pretty hard to squeeze it through a capillary. So the solution, get rid of the nucleus. You don't need it in a blood cell anyway.
Just don't have it.
So they have secondarily, they have evolved anucleate blood cells to go through the capillaries to get to the places where they have to go, solving the problem that the genome presents. Astonishing. Okay, cool. Blood cells, we know about it. We've got them ourselves. Also might be a voucher of our own previous history of having been miniaturized. Who knows? But wasps.
Uh-oh.
Okay, so we have to leave the vertebrate train. First of all, wasps are cheaters and barely count when it comes to miniaturization. And that's because as soon as you outsource a part of your development, it's not fair anymore. Right?
Yeah, you're cheating.
Exactly. And this is why I don't really like to talk about parasites, because parasites, when you outsource your source of energy, your locomotion, most of those things, of course you can get small. If you don't have to make your own heat or make your own stuff, it just floats in the soup towards you, then Plasmodium is the way to go. Be a malaria.
Just be a cell at that point.
Exactly.
Just exist as a single cell.
Yeah, and we see that, right? We see these things that are just like crustaceans. Smallest crustaceans in the world are probably just parasites. They're like, I don't need an adult form. I'll go into the myofauna as a larva to disperse, and then I'll go and lock onto something, go into its body, and I'll just be inside. Okay. Wasps. Right. The smallest wasps have anucleate neurons, which should not be possible.
Because what more is there to it?
And neurons are so complex and so important to consistently be producing proteins to make them function. What is a neuron except for various different ion channels that are suspended in a membrane? You have to have those bits, and they need to be made, and they need to be made regularly. But what has happened is that these parasitoid wasps have, as larvae, nucleate brain cells. And then they undergo metamorphosis, and as part of the metamorphosis, the nucleate brain cells eject their nuclei.
Oh, no.
They've made all the proteins they need.
They're like, I'm done with this.
They only have to live for a few hours.
Who among us hasn't tossed out the instruction booklet once you've built the darn thing? You've built it. Well, I'm never going to need these instructions again. Why would you keep the manual? Get rid of it.
Exactly. Yeah, it's just weighing you down.
My husband would be horrified. He has all of our manuals and all of our instruction booklets. He would be just like
I have
having
all of
an aneurysm.
the Lego instruction booklets from my childhood as well, so I couldn't. But what a fundamental thing to get rid of. What do we understand about neurons? They should have a nucleus somewhere in them, and these ones just don't.
Like, turns out you don't need one.
Turns out that if you only live for a very brief time and you make enough of the proteins to begin with, you can just eject them and you can have a smaller space. And so again, like this is a way that these miniaturized organisms can prove to us what is the fundamental limit. How small can you get? How few cells can you be in order to make things? And then always it comes up of like, what's the value of your research? Why would anybody want to sell this? Which, I absolutely detest that because
Ant-Man would be very interested
exactly.
in this research.
That's actually basically the thing I said. I'm like, well, we're talking about synthetic biology all the time, talking about like synthetic organisms. And how important is it for us to know which of the neurons that you need and which ones you don't? Okay, they're going to come up with C. elegans as their thing because we know exactly which cells you need and which ones you don't. But still, it's important for us to know. And C. elegans, arguably a miniaturized worm. So yeah, I think that these are like, there's a fundamental understanding there that's really, really, really important and exciting. And that is my incredibly long-winded answer to your opening question.
What is a conversation? But
I told you I was a rambler. So.
A question with a long-winded answer. But I mean, we have a tendency to sometimes focus more on the world in macro, like
for sure,
looking more at like the animals that are big and that impress us because they're easy to look at.
And we forget that we are megafauna.
Yeah,
we're
megafauna. So much bigger than most things
by several orders of magnitude.
We can really sell ourselves short by not like.
We are so much bigger than most things. Yeah.
Yeah. We focus so much on the animals that are bigger than us.
Yeah.
That we don't realize that it's not that many of them.
It's so few of them. It's so few. I also got this question asked to me before the review for my big grant. They were like, why are you only doing this end of the spectrum? I was like, how many times have you watched a documentary about blue whales? How many times have you watched one about like thrips? Nobody talks about the hyper-miniaturized things. We're so big that our entire body is full of organisms. Like, ridiculous.
There's whole ecosystems in there.
Ecosystems. You are an ecosystem.
You contain worlds.
And you could almost have frogs living on you. They're so small.
Also, when we think about the world in miniature, we think about tiny critters. Usually that's just bugs, right? Like, We're like, that's bug world. We don't need to worry about what bugs have going on. Bugs got their own thing going on down there.
Secrets. Drums in the deep.
It is interesting to think about like creatures that are much more closely related to us than we might think, right? Like our own vertebrate cousins that are crawling around in the leaf litter.
Yes, and I think they're so compelling to us. Especially, I have a 3-year-old son, and so I get to watch the process of learning about life on Earth again, which is delightful. And I think that there is this kind of intrinsic fascination with extremes, with bigs and smalls. And, I picked up some of the books that I had as a kid, and some of them feature like, this is the largest blue whale. And it's just like a tiny picture in a book because the book is very small. And then they're like, this is the smallest monkey and it's life size. It's like, that's very compelling. And I think that there's such a compelling story about so many of these things. So
we like animals that make us feel small, but we need to be reminded, I think, sometimes that we are big. Definitely.
And that the vast majority of life on Earth has been unbelievably small through time. The fossil record is biased toward large animals because we find them easier and they fossilize more easily. So yeah, we have this preconception of larger animals being like the big players, when in reality we all know what happens when the bees stop flying in the fields.
That's true. It is that like, what is it? The trophic pyramid, right?
Yes.
Like you need a foundation of the ecosystem, and that's going to be the tiny guys.
Yeah. And when you miniaturize, you move yourself. This is really well established. You actually move your trophic position. you're no longer toward those like big consumers. You can move yourself down the ladder because suddenly you as a small frog are food for a big frog. You add a little step in between.
It's frogs all the way down.
It's frogs all the way down. I should mention the smallest tortoise in the world are these padlopers from South Africa, and they're fairly large at about 12 centimeters, but still, that's a pretty small tortoise.
That's a little tortoise.
Delightful. Yes.
I do think there's a charisma to a tiny animal.
For sure.
To like a miniature animal. There is a charisma to that. Like,
there's a reason that book nooks are making a bang right now. Everybody loves a tiny library in their library.
We love a tiny thing. We love a big thing. You make it little. We love that. Humans love that.
Tamagotchi. Like, come on. Pokemon. Literal pocket monsters.
Although I've gone in the wrong direction on my desk, I have little things made big. I have the Lego insect
Excellent.
set. We have made small things big on my desk.
As a resident of Denmark, I have to approve.
Is Lego like huge there?
Lego is Danish.
That makes a lot of sense then. I would imagine they would embrace that as a
Very much
cultural
so.
legacy.
Oh, yes. There's just so much to explore, and I think the more you pay attention to it, one of the greatest delights in raising my son has been to lay down with him at the edge of our deck and just look at what's happening in the grass. Because you can see, you watch the interactions between the ants and the spiders and the springtails and all the little creatures coming out and the small plants that are there. And there's just so much exciting stuff that we really do not pay attention to. So you can cultivate a teeny tiny garden of tiny mosses and stuff. There's just exciting things that are happening at those small scales that it's too easy to overlook, really.
A lot of times I hear people say, like, what happened to roly-polies? Like, you never see roly-polies anymore. Like, we used to see them as kids, and now I haven't seen a roly-poly in years. What, like, implying that, like, something happened to them?
When was the last time you turned over a log?
They're like, they must have, like, gone extinct or they're endangered or something like that. I'm like, just look down. Like, you know why you found them all the time as a kid? Because you were 2 feet tall.
Yes.
You were
It's so
right
true.
there.
It's so true. They are right there. Yeah.
Dig around in the dirt a little bit. You'll find them.
I would say that the isopods are not chief on my list of main concerns for extinction crisis. The detritivores are okay for now.
Everything's coming up isopod.
I want that t-shirt. Sorry, can I have that t-shirt?
Everything's coming up
isopod. Oh, so good.
Next merch design.
It should say Memento Mori, everything's coming up isopod. Amazing.
Okay, last sort of riff that I wanted to go on is I wanted to ask about specifically, because we have mentioned Ant-Man a few times.
Yeah.
And you kind of touched on the idea of were we miniaturized? Did we used to be, did we come from something larger and get smaller?
We know as mammals, yes. As primates, no.
So, yeah, because we would have gone the other direction, but
Yeah.
I guess more in the grand scheme of mammal evolution, getting smaller. And then, like you mentioned, losing traits as you get smaller, having to reinvent them as you get bigger again. Do you have any takeaways or thoughts about miniaturization as it applies to humans? Because I keep thinking about Ant-Man, and I'm like, if Ant-Man turned really, really tiny, I feel like all his organs would stop working.
That's exactly. He would die.
All I can think of every time I watch those movies. I'm like, you would be dead immediately.
He would die of hydration stress so fast.
Instantly, you're dead.
Instantly. Because he shrank in proportion to his body. If he would shrink and become more spherical, it wouldn't be as
That
extreme.
would be great.
But because he retained, relative to his volume, the same surface area, he would lose all of the water in his body unbelievably fast through his skin. Also, his bones would probably disintegrate, and he wouldn't be able to move his face. There's lots of different bad things would happen, but it's the suit. It's the suit. Physics becomes really wonky. It doesn't play along when you get that small, and we're used to big physics. When you get that small, there's a reason ants are proportional to the body size so strong. It's because of the way that the contractile forces act on their muscles and whatever. So yeah, you might be relatively strong proportional, but you'd be so dead, it wouldn't matter. So,
can't act on it.
Yeah.
What are you lifting? Nothing.
There's been. I definitely don't want to end on a super downer. I had some people bring up oh, do you think there's a future in pursuing smaller people? If people were substantially smaller, we would be able to take up less space on planet Earth. You wouldn't have to have ceilings so high. You could have more stacked buildings, blah, 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 blah. And that gets so close to eugenics so fast that I like to very strongly backpedal away from that. I think there are very valuable applications to us understanding the fundamental limits of the body that we have, of the vertebrate body. And I think there are probably valuable understandings from an embryological point of view that we can get from studying some of these things, because we can understand, okay, how do you solve this problem if you have an embryo that has, too few cells or whatever, like what programs can be taken that can allow for the innovations that would be required in order to rescue that, or like, I'm sure that there are very valuable applications, but I, as a taxonomist, fundamentally, I'm deeply disinterested in people. And I don't think there is any sense in us talking about the research that we do because we're passionate and interested and it's exciting and it's fascinating. And why? Why is it like this? We don't know. Just the curiosity alone. We are incentivized as researchers at my university and many universities across the world to pursue curiosity. And then every time we talk about curiosity, they're like, okay, but could it be used by the military? And I'm like, deeply, I don't care. And I don't want to know. What I want to know is, why aren't my frogs smaller than they are? Is there a chance that I can find one that's smaller than that? And how did, Brachycephalus pulex get so small? And what has happened along the way? And what would that mean? Okay, let's abstract. If everybody's talking about space exploration, which again, deeply disinteresting to me, because 100% of all the life in the universe that we are aware of is on this planet.
There's no frogs out there.
Just vastly uninteresting space for light years around us. And until they solve that problem, I'm not looking at the sky. It's the deep sea and the rainforest.
You show me space frogs and then we'll talk.
Exactly. So I don't remember where that was going, but I'll just like the point is just to say that I think these questions have such merit in their own right, because I get the same questions from professors as I get from third graders. Same questions. That means we care as a species about the answer. Not how it matters, not how it affects us, not how we can use it, not how we can militarize it, but it's just interesting. We're very curious.
Human curiosity, yeah.
And we create beautiful stories. I'm pointing at my bookshelf. We create beautiful stories for the sole justification of reading beautiful stories. We started off our conversation talking about Tolkien, and it's just beautiful. It doesn't have any purpose. It doesn't have any value other than the beauty of the thing. Why can we not do research that is simply fascinating and beautiful, and then hang what it actually means for the rest of everyone? Come on.
Maybe the beautiful story is that one time there was a frog, and then it got really small.
Yeah. We are the universe's opportunity to understand itself. Why are we wasting it on doing things that are like, oh, it's going to get me the big bucks, or, oh, it'll allow me to kill more people?
Right. How will this make me more money?
Yeah. So deeply uninterested in those things. What I really care about is why so small?
Why are things small?
Why? So small.
Unfathomably small.
Unfathomably small. I had to dissect one of these small frogs.
How? Literally how?
Yeah, without a microscope. And I can tell you, they are smaller on the inside than you would think. They are
somehow an opposite TARDIS.
It was actually Mini Mum that I was dissecting. They are so small.
Did you have to use one of those lasers that does surgery on a grape?
I did it by hand.
I would never.
I've dissected many, many frogs, so I have a very good understanding of frog anatomy, thankfully. But it was not easy. There was not a lot of tissue.
I would have been recruiting a laser for that. I'd be like, please don't trust my hand with this.
If they would let me take the frogs live out of Madagascar, I would do it with the laser, but they won't, so got to do it in the field.
My thought process in bringing up how could the concept of miniaturization apply to I was thinking our natural history, what traits might have arisen in us that maybe were things that we had to re-innovate, like you were talking about, traits that we may be lost in becoming smaller as mammals that as as we got bigger again, that we maybe benefited from having to like rebuild some structures from scratch.
I do think that there was some of that involved, other than the ear thing. Like that would have been one thing that was happening alongside many others. We would see probably some organ restructuring, organ reorganization as a part of that as well. I know that in another tangent, you may be aware of the work of Stephen Jay Gould, who is a very famous evolutionary biologist. He also wrote for the general public. He wrote a series of essays for the general public on evolutionary biology topics and general learning things. Fascinating person. He wrote a series of essays called Hen's Teeth and Horse's Toes, I believe. And for whatever reason, my father took that out from the library when I was small, and I listened to it on cassette many times. So I kind of grew up listening to the work of Stephen Jay Gould, never thinking I would ever encounter him again. And then a few years ago, I started increasing references to works of Stephen Jay Gould, who it turns out did incredibly important synthetic work on big relevant theories about ontogeny and evolution and these things. So this thing, like these essays that I grew up listening to about like that taught me the wonders of convergent evolution and innovation in evolutionary history, also taught me about like ichneumoniid wasps and the horrors that they incur on caterpillars. It turns out that Stephen Jay Gould is a very, very, very important person in specifically this field, which is just a hilarious coincidence. Anyway, that aside, Stephen Jay Gould did he wrote a book, I think, called Ontogeny and Phylogeny, which gets into some of the early work that talks about some of this stuff. It also talks about, changes in developmental timing and pedomorphosis, neoteny, those terms that we talked about before. And amongst that, I believe there's also discussion about some of these things about humans, because humans have abnormally large heads. Apes in general have relatively large heads, but humans have like particularly large heads, especially when they're very small. And the suggestion there is that this is a change in developmental timing, that the maturation of the head is shifted forward through this process called heterochrony, which is just the changing in timing of developmental events, to allow us basically to have the brain capacity to develop those important skills very early on. And then, of course, if you were to look at a baby scaled up to adult size, it would be absolutely horrific, and vice versa. And that kind of is attesting the fact that there is some level of heterochrony that has happened in humans, but it's not really, because heterochrony is not necessarily tied to miniaturization, but can happen kind of connected with miniaturization. It's not really relevant, right? We are not miniaturized. We have some features that we might say are precocious developmental traits, or you might use other words along those lines, but we are far from small, let's say. We are some of the largest primates ever to exist, although some of the giant lemurs would really knock us out of the water. But certainly we are very, very large, and we don't owe much to a tiny primate ancestor. Although it's worth mentioning that the ancestral primate, when you go back to the root of the primates, especially when you go to, toward the lemurs, if the lemurs, galagos, bush babies, those are pretty small. And in fact, of course, the smallest primate in the world is a mouse lemur from Madagascar.
Madagascar is playing the hits.
Oh yeah, for sure. Yeah, it's a shame we don't have any hummingbirds, because you could really knock it out. But in any case, you can go and study those miniaturized or those relatively small primates and understand a lot about, again, what does it mean to be a primate? What are the fundamental limits of being a primate? But we definitely see some stuff that is very different in those organisms than we see in what was the probable ancestor of our group. So it's unlikely that they were like a hyper-miniaturized primate that led to us. I don't think we have enough innovations between us and the chimps for that to be realistic. I mean, we're so closely related to chimps, it's actually ridiculous.
Yeah, we're not that special.
Yeah, no, exactly. And that is also where it would have had to be, right? It would have had to be between us and chimps, and there's no way. It's just not possible. And we know that several of our closely related homos were actually substantially larger, at least larger boned than we are. So
I did Google mouse lemur.
Oh yeah,
that's a good Google. That's a good image search.
It's a good Google. Apparently, if you add the tag -u to your Google search, it no longer includes AI.
Oh.
which is pretty useful. Although I haven't tested that myself, so just take it at that was the Blue Sky tip that I got.
I use the search engine Ecosia.
Yeah, Ecosia is a good way to do it.
And Ecosia seems to not give me AI stuff
Yeah,
at the beginning.
it's certainly better. I mean, in the fourth row, even with the -u tag, I still see some definite AI products, but
Just
you know,
give me peace, please. Please
Yeah.
give me peace. I would love it. I would love to not have to filter out AI
It's
results.
so
That'd be
bad.
awesome.
Anyway, mouse lemurs are definitely worth Googling. Excellent creatures.
For people who are charmed by the world of the tiny and fascinated, not just by the world of the tiny, but the world of amphibians and the world of reptiles, and would like to hear you talk about them more, could you let people listening know where they can go next, where they can find more of your work, more of your voice?
Definitely. So I have a podcast which is similarly rambly to this one. To be just now, not to you. You're brilliant. Much less rambly than I, although you've maybe just given me the space to ramble.
I'm taking the back seat.
Which is squamatespod.com. You can find us. We're on the main streaming services, so Spotify and Apple Podcasts. Squamates Pod is me and Ethan Kosak and Gabriel Ugetto and Harold Nike. We're all interested in reptiles and amphibians. We try to talk about some of the hot recent news in reptile and amphibian research. We all also talk pretty regularly about what's going on in our lives, and especially, kind of recent research that we've been doing. So Harold's about to finish her PhD, or has submitted and is waiting for the results of her PhD. And I talk about moving to Copenhagen to become the curator a few years ago and those kinds of things. So yes, you can find us there. You can also find me at Mark Scherz, MarkScherz, everywhere on the internet, including Tumblr, which is probably my most active platform, because if there was ever a social media platform where frogs are most at home, it is Tumblr. So you'll find me there. You'll also find me on Blue Sky. I don't really go onto the bird website anymore. And I had to remove some apps from my phone because I had a social media addiction. I also have a YouTube show where I narrate dissections of things. If you're interested in learning how to dissect organisms, you can go to Anatomy Insights on YouTube, and you can get a walkthrough of how to do some basic university level dissections, which is another random tangent thing that I do. You can go to markscherz.com to find out about things that are happening with me and in my lab. You can see trip reports from my fieldwork and those kinds of things. And yeah, if you have any questions, you can shoot them to me on Blue Sky.
Perfect. Mark, thank you so much for your time and for your wisdom and for telling us all about the world of the tiny, for helping us take a magnifying glass down to the leaf litter and take a look at the tiny folks among us. We've had a great time, and we will talk to you later.
It has been a great pleasure. Thanks so much for having me.
Thanks, Mark. Bye. Thank you all so much for listening. I hope that teeny tiny frogs and reptiles and all manner of miniature critters have taken up space in the leaf litter of your heart. If you liked what you heard, I hope you leave behind some kind words for us in a review on your podcast app of choice. If you'd like to hang out with us online, we're on Facebook, Instagram, Discord, Blue Sky. Links to everything will be in the episode description, along with all those links to Mark's work. You can also send me an email at ellen@justthezooofus.com if you have a cool animal you'd like to hear us talk about on the show. We would like to thank Maximum Fun for having us on their network alongside the other amazing shows like the ones you heard promos for here today. You can go check those out, learn more about the network and how you can be a part of supporting our show over at MaximumFun.org. Finally, we'd like to thank Louis Zong for our theme music. That's all for today. We'll see you next week. Thanks. Bye.
Maximum Fun,
a worker-owned network
of artist-owned shows.
supported
Directly
by you.